All right, welcome and thank you everybody for joining our Sargassum webinar series. We are launching this series to raise awareness about key issues surrounding sargassum. Each webinar will highlight different aspects of sargassum research, management, and its impacts on coastal communities and ecosystems, as pictured here in this graphic. The goal of the series is to foster knowledge sharing and regional collaboration across affected areas as we continue to see the impacts from sargassum inundation events. My name is Catherine Egan. I'm the Southeast and Caribbean Regional Coordinator for NOAA based in Key West, Florida, and one of the hosts of this webinar series. I help co-lead and coordinate for NOAA's Southeast and Caribbean Regional Collaboration Team, or SECART. SECART is one of 8 regional collaboration teams across the country, working with both NOAA staff and partners throughout the United States to foster stronger collaborative ties, all in an effort to improve NOAA's regional capabilities and services. You can find more information on SECART by following the link that we'll put in the chat in just a moment. So here were the webinars that took place during our series, and today is our 5th and final webinar. You can scan the QR code at the top, and that will take you to the website where you can view descriptions of these past webinars. Tomorrow or Monday, all of the recordings from our webinar series will be up on this website as well, and we'll put a link to that in the chat. All right, and with that, I'd like to introduce my co-hosts. So with me today are Max Brown and Josie Galloway with NOAA's National Centers for Coastal Ocean Science here at NOAA, or NCOS. And I'll pass it off to Max to introduce himself.
Hey, thanks everyone for coming again. I'm Max. I'm a contractor working with NCOS, National Centers for Coastal Ocean Science, as a program analyst. I have a background in coastal ecology and phytoplankton. I started working on sargassum management a little over a year and a half ago, and helped pull together an assessment of the federal policy landscape. Following that, I helped organize and convene a research workshop, sort of trying to figure out next steps for helping out and supporting sargassum management, which sort of fed right into this series. And Josie helped on a lot of that work. Pass it off to Josie.
Thanks, Max. Hi, everyone. I'm Josie, and in my role, I serve as the Executive Secretary for the Interagency Working Group on Harmful Algal Blooms and Hypoxia Research and Control Act, which sits under the National Science and Technology Council. In this role, I help to coordinate with federal agencies to develop strategies to research, prevent, control, and mitigate the impacts of HABs, which include sargassum. Over the past year, as Max has said, I've helped work with interagency scientists on the development of the policy analysis and on workshops and webinar series like this. If we could go to the next slide, please. All right, so I'll go over some housekeeping items. So first, all participants will be muted during this presentation, but you can send us your questions in the questions box. Second, due to a large number of participants, some of the slides might be a little bit delayed. And lastly, we are recording this webinar and it will be posted to our website after the series ends. And with that, I'll pass it back to Catherine to introduce our speakers for today.
Thanks so much, Josie. Thank you, Max. Ike, I'm going to go ahead and move screen sharing permissions over to you and let you get set up. And while Ike is getting organized, I'll go ahead and introduce our 2 speakers today. So we're really pleased to have Dr. Bradley Whalen, Group Lead of Biological Processing at the Idaho National Laboratory, and Dr. Ira Levine, Professor Emeritus and CEO of the Algae Foundation. So I'll introduce Brad first. So Dr. Brad Whalen has been developing biomass pre-processing approaches within the Feedstock Technologies Department at the Idaho National Laboratory for the past 11 years. His focus has included enzyme technologies to improve biomass processing, storage approaches for agricultural residues, ash reduction in both microalgae and seaweed, and preservation techniques for stabilizing wet feedstocks like micro and macroalgae. And we also have Dr. Levine, who was awarded the 2016-2017 U.S. State Department's Distinguished Chair Fulbright and the 2009-2010 Fulbright New Century Scholar. His 45 years of algae farming and cultivar enhancement experience includes work in Canada, Chile, China, Fiji, India, Indonesia, Italy, Japan, Malaysia, the Philippines, and the USA. Dr. Levine has authored 36 publications, 105 presentations in 46 countries, 4 book chapters, 3 books, and 29 grants totaling over $10 million. So thank you both so much for being here today and presenting. Ike, I just want to let you know we can see your screen. It looks great, and I'll go ahead and pass it off to you to begin.
Well, good afternoon, everyone. And it's a pleasure to be here. And for the last of the 5 webinars, Brad and I hopefully will take you through where the sargassum is landing, why it's there, how come there's so much, what can you do with it, and Brad's work is essential in terms of how do you stabilize seaweeds that land all throughout the Caribbean and be able to centralize it to make commercial products out of it. But from the first slide, you could see that sargassum is not a new issue. The Sargasso Sea, the myth, the lore, the legends have been around for over 500 years. And so we'll have a little fun with it, and hopefully you'll learn from it. If you have any questions, put it in the chat or wherever you put questions, and we'll answer as much as we can. And if you have any questions after the fact, you can always reach me through the Algae Foundation, where we work on workforce development, educational training for the algal commercial sector. Okay, so let's proceed. So in terms of the Sargasso Sea, we've all grown up with the creature from the Black Lagoon, the lore of lost ships, lost airplanes. Again, it's been a mystery between Bermuda, the North Carolina coast, and Cuba. And so there's, we all grew up with these type of comics. Well, perhaps you have to be my age to grow up with these kind of comics. But the point is, the Sargasso Sea has been the source of legend and stories of which has kept our attention to the Caribbean Sea. But as you can see here in the first and second webinars, they discussed, the origin of the Sargassum Belt, perhaps coming from the Sargasso Sea. The Sargasso Sea is, again, a central Atlantic gyre that's been there for quite some time. And you're looking at a group of asexual seaweed that hasn't had sex in a couple 100,000 years. And so with that said, how did it break out? How did it go from a secluded or contained group of organisms to this massive biological phenomenon that goes from the mouth of the Mississippi River to the west coast of Africa? And I will say recently in offering lectures on luxury ships that we went through the west coast of Africa. And I've also done one off the coast of Brazil and through the Panama Canal. And the Sargassum Belt was visible and prominent in both cruises. So it was really quite, joyful is the wrong word, but it was reassuring that what we say in these lectures are actually happening out in the ocean. So let's go to the next one. The question is, why so much? And people don't realize, if you're talking 30 to 50 million metric tons of biomass, that's a heck of a lot of biomass. And so who's feeding it? Well, originally, a lot came from the Mississippi River and the nutrients in the Gulf of Mexico certainly came from there. But that's not what's driving this this golden or brown tide. And so it's really the over 200 million cows that now occupy the Amazon River basin. And what are we talking about? We're talking about 4 billion tons of wastes, and that, both liquid waste, urine, and manure. And 1%, just 1% of that massive output from the 200 million cows is nitrogen. So you're talking 42 million tons of miracle grow that's coming out of the Amazon River. So in terms of where these 40 million tons of nitrogen affect, these are just listings of the landings of sargassum. And so essentially, it's everywhere. Almost no island has been immune. No Central American or northern South American country has been able to avoid it. There are no barriers that will, totally keep them off of the shores. And so this represents a near global, at least a regional phenomenon that really needs to be discussed and negotiated, because each of these countries have different regulations on what you can and can't do with the biomass. So let's take a look at sargassum. You can, I was going to say, blame the young man. He's my age, Dr. Brian Lapointe, excuse me. Brian is in charge of the discharge of nitrogen on the east coast of Florida. So he's worked with seaweeds longer than I have. So he's over 50 years, and he's worked with trying to mitigate or minimize the nitrogen from the sugar beet and agricultural runoff from the state of Florida into the Indian River Lagoon system. And so, as you can see, a little playfulness with the bloom in the Florida coastline. But as you could see here, in terms of whether it's the Caymans or Puerto Rico, we're talking about more biomass than you could than you're than you can perceive normally. And so, in China, they have a seaweed hab in terms of ova up by Qingdao, which affected the Beijing Olympics several decades ago. But this is a mass that your mind and eyes have a real difficulty in putting their arms around. Well, again, this is a dynamic phenomenon, and as such, from the satellite system and computer programming that comes out of the University of South Florida, they have determined that the belt itself is shifting. So again, we're talking about, what is it, 2011, that it became, the perception of it as a unified system was reported. But it's shifting northward, and we're looking at a shift of around 20 degrees in terms of, instead of south of Cuba, we're talking north of Cuba. And so this is a significant departure, and it's a function of wind patterns and surface currents. And so again, this is this each 1 degree shift alleviates the landings in certain areas, but then again, maximize and shifts it to new areas. So in this respect, it's something to keep quite a close eye on. So in terms of what does it mean? Again, if you own a restaurant, a bar, or a hotel on the beach, and the beach is starting to smell like rotten eggs, you're not a happy camper. So the question is, what are you going to do, and how are you going to do it? Remains not a mystery, but it remains a significant challenge that over the course of 15 years really hasn't been solved. And so, again, you can have human muscle, as you see here, just sheer pitchfork and manpower or human power to physically remove the biomass after it's landed on the beach. Here we have a little bit of mechanization, but it's still, again, physical labor to remove it from the beach landing. And you're talking about, if you're trying to prevent the harmful release of sulfur compounds, and the aerialization of sulfur compounds with all of its potential effects on human health, you're talking about 48 hours. And so there's nothing more uncomfortable than being in rotten brown algae. Uh, Worse being brown versus the greens and reds. Why? Because they have more sulfur compounds. And so if you look here, again, organized parties to remove the biomass. Now, what are they going to do with the biomass? Just like in China when the army removed, what did they say, 18 million pounds of the ova, it went to a landfill. What a waste of potential opportunity. And although in the beginning these piles of seaweed were removed to landfills, over the course of the last decade, what to do with it has certainly become a dynamic source of research and development. But here in Galveston Bay, we're using heavy machinery. You think, of course, you're going to use heavy machinery. It's certainly more efficient than hiring scores of day laborers to do this. Unfortunately, and especially in Puerto Rico, they've, I believe, outlawed it because of the weight of these heavy machinery compacts the soil, damages potential reproductive zones for turtles, and adds to beach erosion. So heavy machinery may not be the answer. So then the question is, why not just get it before it comes ashore? And that makes a lot of sense, except for the fact that in certain waters it's illegal. Why is it illegal? Because these living reefs, these are like floating rainforests, are the husbandry and habitats for critical species. And so, oddly enough, you got to wait till it lands and those critical species would die, but you can't, in many places, use offshore boats. Plus, the capital cost of building these new facilities. So the question is, what can you use it for? And so this certainly says you can use sargassum for almost everything. Can it be a biofuel? It could be bioethanol. Can it bioremediate? Well, you look at those 40 million tons of nitrogen. It's certainly stripping out a tremendous amount of that. So in terms of bioremediation or phycoremediation, certainly it's a valuable asset. But if you look all through this circle in terms of biodegradable bioplastics, construction material, which we'll go into in a second, paper fibers. But we've also met last month, had the opportunity to meet a fashion designer who does the gowns for the Met Gala. She is using algal-based fibers and algal-based material, not only as the core material in a high fashion, but also the dyes in making spectacular prints. And so in terms of footwear, the development of biodegradable beach shoes. I can't tell you in Micronesia, in the Atoll, how much plastic on uninhabited islands there are. Not only the 2 liter bottles, but a tremendous amount of footwear and other single-use plastics. So in terms of pharmaceuticals, the browns and the active compounds in terms of whether they're antiviral, anti-tumor, or antibiotic type of compounds. This list is both comprehensive and broad, and we'll have examples of individual efforts moving forward. But in terms of what is the totality of material that you can glean from sargassum, so if you take a wet ton, and this is a metric ton, if you take a wet ton, this gives you an idea of how much material. Not all of this comes from 1 ton. Each one of these figures that you see, for example, over 2,000 pairs of shoes can be made from a single ton of fresh sargassum. And so, again, people say, wow, that's a tremendous amount to get all of that stuff in one slide from 1 ton. Please don't. That's not the case. And so, again, we'll take a look at a few of these applications. But again, whether you use it fresh, extract the proteins, the carbohydrates, the lipids, although not a lot of lipids. a tremendous amount of vitamins and micronutrients and minerals come from this. Fucoidin, for instance, is a very active compound. And so the other thing is you see sheep's, just to give you an anecdote, during the potato famine in Ireland, where they had 6 million people, 2 million immigrated to the United States, 2 million survived, and 2 million didn't survive. But the coastal communities had significantly reduced hunger issues because they incorporated fresh harvested seaweed, both from the shoreline for both human consumption, but also animal consumption. So in terms of one of the things you can do is dried either kelps or sargassum can be used as a partial substitute in the development of construction material. And so if you're looking at a third replacement for concrete, concrete being one of the top 5 largest emitters of carbon, this is a significant reduction in the carbon footprint of the construction world. So 3,000 bricks a day is not insignificant. The question is supply, and that is the big challenge, and that's something Brad will talk about in just a few minutes. Other things that are being done, and these are easy. Again, these are primary processing efforts in terms of air drying on the coast, whether on the ground or elevated by chicken wire frames. Seaweeds tend to stabilize when dry. Again, they can be used both as you see here, fertilizers, soil elicitors, soil amendments. And so sargassum is located all over the world, and so it has been formulated into products all over the world. And in terms of some of the others, these products don't all come from sargassum, but you have biodegradable footwear, biodegradable flip-flops. You have a substitute of petroleum-based foam for seaweed-based surfboards. And in the bottom left, again, a time series of the biodegradability of plastic bottles. So these are an amazing array. And as the biomass becomes more available and more stabilized, the commercial aspects will only grow once industry knows that they have a recognizable and affordable source of consistent algal-based biomass. And so I'm going to introduce just the last couple slides that I have. The algae prize, it's been supported by the U.S. Department of Energy. We've had 2 of them, the algae prize 2023 through 2025, just crowned its grand champion last month at the National Renewable Energy Lab. And this example is from Algae Prize 1. This is from one of the Puerto Rican teams that made the top 5. They became a champion of the Algae Prize 1. And here you see sargassum being dried, and they converted into 3D printing filaments. And as you can see here, they made different types of outputs, one of which, in terms of commercial applications, are algal-based covers or cases for your iPhones. And so here's the team from Polytechnical University of Puerto Rico. They received an award of first of initial of 10,000, and for becoming one of the 5 champion teams, an additional 10,000 for Algae Prize 1. Just last week, the University of Puerto Rico, their team was developing, and in the far left-hand corner, they're developing both biosensors and better lithium batteries. And they're finding algae nanomaterials are tremendously helpful, and sargassum is a pretty darn good transmitter of electrical or electrons. And so that's been a tremendous source of interest. And just from the presentation at the National Lab and the social media notifying their award, commercial interests have contacted the algal-based team to commercialize some of their work with sargassum. So that's unbelievably exciting. But what good is this if you're not trying to, in terms of reaching out to the community and bringing them into this opportunity? And so University of Puerto Rico and their team, Algal Nano Plus, have really, really gone way above the Algae Prize requirements of both social media, but also hands-on reaching out to young people. And some of their presentations are just knock you out. In the lower left-hand corner, you'll see sort of looks like an oak leaf with green lines. Well, those green lines are algal-based materials that are actually carrying electricity, and they light bulbs. They light bulbs from it, from the sargassum extracts. And so it's really, really exciting stuff. And the fact that each one of our 15 finalist teams are doing this all over the country is a remarkable testament to the power of algae and the excitement that algae brings to the general public. So I think that's my last slide, so I'll turn it over. And if you have any questions, we'll have plenty of time at the end. Thank you very much. Brad, take it away.
Hi, everybody. I'm Brad Whalen. You might be wondering why someone from Idaho was invited to this seminar. We're pretty distant from the coast, but I'll explain why that is. Next slide. So at Idaho National Laboratory, we have the Biomass Feedstock National User Facility. This is a congressionally designated facility that is utilized to overcome some of the challenges with taking waste materials like sargassum in this case, or other forest residues or ag residues, and how do you get them prepared so that they can be converted into useful items. And so that's the purpose of this facility. And as you can see in this picture, there's a lot of machinery. And so we have multiple scales. We can do process development on a bench scale all the way up to 5 ton per hour scale. And so that's kind of, the purpose of having me involved in Sargassum project is to help develop the processes to get it into the marketplace. Next slide. This graphic just kind of gives an overview of what we try to achieve in our terrestrial biomass research, where we take these residues from disparate sources, be it the forest or ag fields, and we learn all the processes needed to develop a feedstock that can then go on to conversion. And that can be, how do you make it flow better? How do you get the particle size just right so it converts well? Or maybe your particular feedstock is recalcitrant. What kind of conditioning can you do to make that more convertible and a better feedstock? Maybe there's different parts of the plant that are better for one purpose than another. We study how do you separate these materials and fractionate them to achieve different goals. And we also look at how do you bring different materials together so that they meet the specification for a given conversion product. So we blend things together, and then we look at how do you densify that material so that when you're shipping it to a distant, conversion facility, how do you minimize those transportation costs? Next slide. And we recently went through an upgrade where traditionally we've worked primarily with things like corn stover and forest product residues. We've added more equipment to our facility that enables us to work with wet materials like sargassum, and so we can start to understand how to best utilize these resources. Next. And so whether we're at the Department of Energy lab, and so our end focus is energy uses. It's not to say that we aren't aware of various other co-products that could be made, but our end focus is for energy. So as I'm developing this seaweed feedstock system, we have in mind two different outlets for this material. One being fermentation to fuel, so you acquire the sugars, ferment to get your fuels, or use temperature and pressure in a process called hydrothermal liquefaction to produce a bio-oil that can then be upgraded. And so we're focusing on what are the pre-processing steps that are necessary to enable these two pathways. Next slide. And so this is a look at seaweed feedstock logistics. These are different unit operations that are important to take the material from the coast where it's landing on beaches and get it ready for conversion. So all these things that we need to think about. And Ike mentioned, where do you collect it? Do you collect it at shore or at sea? Well, at sea you have ecological impact, and it may not even be legal. And if you're collecting on the beach, you might incorporate sand or something into your material, or if you don't collect it right away, maybe it's degraded a little bit and the quality is no longer good. So those kind of questions all need to be addressed. And how far can you transport this material before you spend more energy transporting it than you could ever get out of the feedstock? And so how far can you do that? And water and ash content of this material is a concern because they're heavy, and that limits how much you can transport in a given truck or whatever. And then how stable is that material in transportation? It's also been discussed how this is a seasonal resource. It's not available year round, so there's some period of time where it's not present. And so if you're going to develop a process and invest in equipment and capital expenses, you need to make sure that you have feedstock available year round. And so you'll need to learn how to store the material and preserve it to prevent degradation. And one of the challenges with sargassum, it's very wet, a lot of energy content, and so it's prone to degradation. And finally, before conversion, you need to look at pre-processing to help the conversion process go well. That's milling, removing contaminants, and before you can even begin to produce something. Next slide. And so we're developing a storage approach, and we're modeling this approach after a technique that's been used for terrestrial biomass called ensiling. And the picture you see on the right is of a silage pile in Iowa of corn stover. And the one on the bottom is a silage tube, just showing you the different configurations that are possible with this technique. And this works on the principle of lactic acid fermentation. So lactic acid bacteria will consume some carbohydrates in your material, produce lactic acid that drives the pH low and preserves the material. However, with sargassum, there's some particular challenges that need to be overcome. It has a high buffering capacity, so it resists pH change. It also has a high moisture content, and that's a difficult environment for lactic acid bacteria to thrive in. And also the sugars are distinct from terrestrial biomass. So you have things like fucoidins that are sulfated carbohydrates, and you have alginic acid, which is uronic acid. And so these are just different from what we typically think about in an ensiling approach. Next slide. So this is a piece of equipment in our facility that we're using. It's a screw press. So it uses a screw to advance material against a backstop, and it squeezes water out essentially. And so go ahead to the next slide. And so we're able to take something that starts at 85% moisture and reduce it down to 60% moisture. And along with that moisture, we're able to reduce some ash content as well. So we can go from 36% ash down to 21%. And that might not be suitable for all conversion processes. So we also looked at, could we reduce it further? So we combined that screw press operation with the acid treatment. And we're able to get the total ash content all the way down to 7%. The picture on the right shows how we're doing our storage studies. So the leftmost jar contains our screw pressed sargassum, and the middle jar is just the sargassum that as received from Puerto Rico. And then the bottle on the right shows our filtrate that comes from the screw press. And so you can see there's a lot of material still in that screw press filtrate, and so we'll need to study a little bit how to best utilize that so we use all the material so we're not wasting anything. And the table on the lower left shows our results in storage. So if we don't treat anything before storage, we see about a 7% loss, and that increases to about 14% over 90 days. But if we first press that material and lower that water content, we can minimize that to 2.5% loss over 90 days. And acid treatment's a little bit improvement over that. Next slide. So what is the ash content being removed? So the untreated is in the green bars here, and you can see, not surprisingly, you have a lot of sodium and a lot of chlorine, but you also have a lot of calcium and magnesium. Now with screw pressing, we can reduce that sodium and chlorine content significantly, but it doesn't really reduce the divalent cations very much. But when we do an acid wash, we see a large reduction in that content. One of the concerns with sargassum is the heavy metal content, particularly arsenic. So you can see that in the table over here to the right. We started with about 120 ppm in our material that we worked with, and we lost about half of that in the screw press operation. So some of that's coming out in the filtrate and will need to be taken care of before it's discarded. And so, just showing that we're trying to follow where the arsenic's going. So that'll be a critical concern as you're developing products. Next slide. So this graphic just shows you, this is approximate composition of our untreated screw pressed and acid wash material. So we're looking at the ash, volatile matter, and fixed carbon. And you see that with screw pressing, we're able to lower the ash. And as a consequence of that, the total amount of volatile matter increases to close to 80% of the material versus where it started at 60%. So we're producing a better quality product that has higher energy versus just straight out of the ocean. Next slide. So this graphic shows what's happening to the sugars. I don't have all the sugars that we followed because it would be very difficult to interpret, but I just put the major ones here. And the first 3 bars in each group is for the untreated, and 30-day storage untreated and 90-day storage untreated. And you can see that the glucan appears to like we're making some glucan in storage, but what's really happening is we're consuming other things, and so that's becoming higher concentration overall. It's proportionally increasing. And so what are we losing in that same untreated storage scenario? And if you look on the next 2 groups on the right, these are uronic acids, part of alginate. And so we're losing quite a bit of the alginate in the storage without treatment. And so if your product is alginic acid, if you don't treat your material before you store, you're going to lose it. And so what's that treatment? Well, we can screw press. That's the magenta item, and then the light green to the right of that. So we basically are preserving that alginic acid in storage just by screw pressing it first. Next slide. So all of this that we've been talking about doesn't really matter unless we have people that can execute this vision, this goal of turning this material into usable products. And so along with Ike from the Algae Foundation and Dr. Cliff Luimi and Dr. Gary Gervais from University of Puerto Rico, we've been putting on a workforce development seminar in Puerto Rico to undergraduate students, and where we start with a lecture to tell them just what we're talking about today. What is that problem with sargassum, and what is its potential? Next slide. And so we take these students out to a field experience. We go to a seaweed farm. We look at what's the latest efforts being done to learn how to cultivate seaweeds. This is in Maguez, the Marine Sciences Center. So we tour their facilities. The students are able to look at algae growing in cultivation barrels. Here they're looking at Gracilaria. Next slide. And then we have workshops with the students. So here on the left, we have Dr. Leva Lawrence from National Renewable Energy Lab talking about their development of a biorefinery to convert sargassum into ethanol and other useful chemicals. And on the right, we have a local company, Carbon Wave, giving a presentation on products that they're producing from Sargassum right there in Puerto Rico. Next slide. We also have the opportunity to give workshops or hands-on workshops to the students where they extract pigments from algae and then use that to, paint banners that express their affection for all things seaweed. And then on the right, we're doing some seaweed pressings where the students are taking the material that they collected on the reef and they're classifying it as to which organism it is, and then using that to create art that they can hang on their wall. Next slide. So in summary, feedstock logistics are critical to realizing the potential of Sargassum biomass. Different pre-processing techniques can increase the quality of biomass and also increase its stability and storage. And just as we just finished discussing, young people in Puerto Rico are really eager to learn how they can get involved and participate in making this vision a reality. And with that, I'd like to thank the folks at INL that helped me with this work, our collaborators at University of Puerto Rico, Dr. Luimi and Dr. Gervais. They've been really instrumental in helping us get biomass and to execute on the workshop. And I'd like to thank Andrea Del Mar Malavi Bonilla. She was an intern with me here at INL and is from Puerto Rico. And of course, I'd like to thank Dr. Levine from the Algae Foundation and his help in doing the workshops and this seminar, and Dr. Loretta Roberson from Marine Biology Lab, and Dr. Lawrence from NREL, and also the company Carbon Wave. And with that, I think we're ready for questions. And I'd like to thank all you for listening. Thanks.
Thank you both so much. That was a fantastic presentation. We do have a lot of questions that have been coming in. I'm happy to kick us off. Let me look over here at our question list. So we had questions that kind of came in throughout the presentation, so I'll try to target ones that may be most applicable to what you both presented on. So Ike, this one came in during your portion of the presentation, kind of when you were going over sargassum blooming at sea, and this person asks, what are the currents like where sargassum blooms most, and can that support ocean farming?
Well, it wouldn't be ocean farming. It'd be more like ocean ranching, because you would be collecting it. That would be the challenge. The problem there is, of course, you'd be collecting a lot of bycatch also. But there are some thoughts to open ocean capture, just like you have open ocean capture of plastics in the Pacific. Again, the cost of which offshore husbandry is not a trivial matter. So you'd have to manage the value of the biomass versus the cost of capturing it.
Mm-hmm.
Thank you. I'll pass it off to Max, if you'd like to do another question.
I guess continuing that, this question is, talking about, because you discussed removing it while it was onshore and then removing it offshore. This question from Vincent is saying, is there a middle ground for removal, such as removal while it's in the nearshore zone? Are there studies on sargassum's ecological value right before it beaches? I can't imagine it being high in that scenario.
Well, you'd be surprised in terms of the amount of diversity of that is found there in terms of both vertebrate and invertebrates. Well, whether fish, shrimp, there are endemic species, endemic eels, shrimp, crabs, and fish that are found nowhere else but within the floating, rainforest, so to speak. And so Has there been a definitive standing stock survey? I'm not quite sure there'd be a mechanism for the invertebrates to leave the biomass, whether it's 10 meters offshore or 100 meters offshore. And so, unfortunately, the demise of the biomass will come about not when they're near shore but when they are onshore and subject to immersion versus immersion. And so when they're subject to rain, fresh water, varying temperatures, and the drying out once they're onshore. So yeah, I think that might cover it.
Great. Josie, do you want to grab the next one?
Yep, yeah. So we had several people ask about the heavy metals and other toxins or contaminants that may be in sargassum. So, and including arsenic, which I know, Brad, you touched on a bit. But can you talk about how those components are removed from sargassum or can be removed from sargassum before it's made into some of these different applications and other materials?
Well, the first thing you want to talk about is where's it coming from? And
Yeah.
so, where is the arsenic? Where's the mercury? Where is it all coming from? And a lot of it's coming from the mining of gold in the Amazon River Basin, not only from corporate, which tend to be more responsible, but there are pirate mining operations that use arsenic and other heavy metals to separate the gold from the ore. So that's unfortunate. So there are surveys over 1,000 miles from the mouth of the Amazon that shows contamination both in birds and turtles from these heavy metals. But algae are the most amazing absorbers of nutrients, heavy metals, and radionuclides. So just as a for instance, any algae that I import from Japan, China, or Korea stops first in a lab in Nevada for radionuclide testing. So any sale of seaweeds that are near Fukushima need to be certified that they're radionuclide free. So if there's mercury, cobalt, molybdenum, any divalent cations in the water, they'll end up in seaweed. Where they're located, some are ephemeral, some are just attached and can be washed off from the external biomass. Some are bound to the cell walls, and some are absorbed internally.
And as far as removal techniques, I don't know if I have, a good answer for that. I have heard anecdotally some people saying that they're able to remove the arsenic and contain it. I don't know how they're doing that, and so I can't really comment on what those processes look like, but it's something to be watching for and as that develops.
Thank you both.
Yeah, thank you both. We'll have to bring you back next year to give us an update on the science there.
There you
That's a
go.
question that we get kind of a lot, and then we've also gotten in
Mhm.
a lot of these past webinars as well. So thank you both. I'm going to go ahead and read off our next question here. So somebody asked, how can a long-term industry develop based on sargassum blooms that shift in both time and space? I don't know who'd like to take that first. Maybe you, Ike?
Well, that's the challenge with any wild harvest, with any sea ranching, so to speak. The consistency of biomass tonnage and quality is the key. So that's why capital infrastructure will be rare or at least limited because of the risk. And so, in terms of all investment, it's about risk and access to the biomass is key. So that's why Brad's work at INL is so essential. If you could stabilize it and around the region, then you'll have at least a 6-month window for collection, and then
Mhm.
perhaps the processing can be drawn out throughout the 12 months. but it has to be stabilized. And that's been done in terms of commercial reds around the world, because most of the carrageenan and agar processing facilities are not in the countries that are farming it. So you're talking about air dry down to 38% wet weight and being able to be stored and shipped around the world. So again, it's but those are commercial reds, not browns, and you have different biochemical challenges. So that's why Brad's work is so essential to creating a commercial opportunity. If you could stabilize it at the source, you can transport it to centralized locations and minimize the factory development
Yeah.
to single source.
Yeah, that's precisely it. I failed to mention that my work is funded by the Bioenergy Technologies Office from the Department of Energy. And one of their focuses is to develop feedstock processing technologies that reduce that risk and encourage investment um, to develop these industries.
Thank you both. I'll pass it off to Max.
I like this question. I'm gonna sort of frame it a little differently, but this question says, as a Kenyan in the U.S. who's worked with Sargassum, how can I tap into the workforce? And then maybe thinking about this more broadly, I guess, how can people sort of tap into, work with Sargassum or sort of start a business potentially?
Well, I mean, again, seaweed has been recognized as an economic development engine for over 50 years. The UN paid for the training starting in Indonesia and the Philippines for eucumoid seaweed farming. So once you have the recognition of an opportunity, you have entrepreneurs and then industrial interest to take advantage of it. So again, in terms of young people getting into it, are they going to be on the biological side, the processing side, the marketing and distribution side? Each one of them are opportunities for young people. And I hate to make this a commercial for the foundation, but the Department of Energy supports ATEC, the Algae Technology Educational Consortium, whose entire focus is workforce development and education and training young people and the different aspects of the algal-based value chain. So whether it's farming, collection, processing, new product development, building a company, all of those are part of the Department of Energy's effort to broaden the participation in the algal blue economy.
And I would add to that, Ike's involvement in the Algae Prize demonstrates just how young people can get involved. You can just start, developing ideas and get your ideas out there. Some of the things that they've come up with have been remarkable, and these are people that have limited experience in algae. So there's not you don't need to wait until you're an expert to try and get involved.
Good point. Yeah, thanks.
I have another question from our chat box that I'd like to ask, and this one's pretty big picture. So, in your view of the many sargassum-derived products and derivatives, which show the greatest commercial potential over the next 5 to 10 years, and which value chains offer the best balance between economic viability and scalable near to medium-term implementation?
I'm only laughing because that's probably a 6 hour conversation. But
Well, we
again,
have 6 minutes.
well, everybody knows me as long-winded, so I'll try to be as brief as possible. In terms of algae, there's always been a dichotomy of its use. You have commodity type usage, whether it's biofuels, biofeed, biofood, and they are big biomass, low unit price value.
Yeah.
Versus, for instance, and this has been a product for many, many years, R-phycoerythrin C-1. Once refined, this red molecule that glows in the dark is worth $60,000 a gram. So, you're looking at, $500 a ton versus $60,000 a gram. So the totality of the algal value chain, which is approaching or exceeding $20 billion now, and that a large in part due to the renaissance of both microalgae and macroalgae research and development supported by the U.S. government. That can't be understated. That in terms of sargassum, probably in its infancy of commercial applications would be the commodity based, whether it's soil amendments, fertilizer, biofeed for animals. Then you get the secondary, a wee bit more mature types of processing, whether separating out the proteins and the carbohydrates and using it for both cosmeceuticals and nutraceuticals to really high-end biotechnology type of applications in terms of biodegradable plastics. And now some of the new work out of San Diego, who won the algae prize #2 from UC San Diego, the ability of algae to create these much higher density plastics and also secrete algal-based compounds that can actually dissolve plastics. And so that came from Steve Mayfield's lab. So you have a tremendous opportunity. It is almost unlimited. And as long as you're going to have cows in the Amazon, you're going to have sargassum in the western and central Atlantic. So I wouldn't worry about saying, "Oh my God, we better hurry up because it might disappear." That's probably the smallest risk. It's being able to safely and conservatively store it while you want to use it, and that's why Brad's work is essential.
Anything to add, Brad, from like the processing perspective and the methods
Yeah,
you're
yeah,
using?
I would say, Ike brought up, either you can have these high value things that are low quantity or commodity scale chemicals like fuels. And our view is you kind of need a mix of both in order for the whole economic paradigm to work. So you need to be, you know, getting high value compounds as you produce fuels, and so that'll go toward developing better processing techniques that enable the collection of both materials.
Thank you. I think we have time for maybe one more question, possibly two, but at least one.
Thanks, Josie. I have to be honest, I think that was probably a good one to end on because we did get some questions that came in that were very specific about your methodology, Brad, and I think maybe we'll
Mhm.
just forward those ones on to
Yeah,
you if
forward
that
them on to me
works.
would be great. That'd be helpful.
Okay.
Thank you.
Yeah, absolutely. Yeah. And if anybody has questions for our speakers, I put our SECART email here in the chat, and you can follow up with us, and we'll make sure your questions get to Ike and Brad should you have any more. Yeah, and I think with that, we'll go ahead and conclude our final webinar for the series. So, Brad, Ike, thank you so much for closing us out on our sargassum journey here for the past 5 weeks, where we've taken our audience all the way from the development of sargassum offshore to what are its uses once it kind of washes up on our beaches here. So we really appreciate you both being here today, and we appreciate everybody for joining us over the past 5 weeks. Links to our YouTube channel and website are all in the chat, so you can catch every webinar there if you missed it. And with that, we'll go ahead and conclude. So thank you, everybody.
Yep. Thanks for
Thank
having me.
you for the opportunity. Cheers to all.